All right, so we are going to get started on chapter 25, which is the history of life on Earth. So we've talked about this idea that evolution includes both macroevolution and microevolution. Macroevolution is generally when we think of changes that happen over very large timescales. And these things will include the emergence of terrestrial vertebrates. So vertebrates, which are animals that have spines that live on the earth, the impact of mass extinctions. So we'll talk about what those examples are. And over to the right here is an example of a dinosaur that was extincted in one of the most important extinction events and well studied, which is the KT extinction. And then we'll talk about the origin of flight in birds. And it is the case that we know from the fossil record that past organisms are sometimes very different from those that are alive now. But we also see that some are very similar. So we'll talk about this. So when we think about what the conditions were on the early Earth that made the origin of life possible, these are some ideas about these chemical and physical processes. So it started with abiotic synthesis of very small organic molecules. So if we think about what abiotic means, A means without, bio is life. So synthesis from non-life forms or joining together of these small organic molecules. These small organic molecules, the origin of which is still debated, and we'll talk about this too. We join these together to put together our macromolecules. That's our list of biological molecules that we've talked a lot about. So things like proteins, RNA, DNA, fats, right? Those are our examples. These macromolecules were packaged into what we would describe as protocells. So this would be a type of pre-cell. And then what we also found was that then we needed to have origin of self-replicating molecules. So things like our DNA and RNA would be examples of those. So the Earth formed about 4.6 billion years ago, along with the rest of the solar system in the Big Bang. And what we found was that after the Earth was formed, the bombardment of the Earth by rocks and ice likely vaporized water and prevented seas from forming before about 4 billion years ago. Excuse me, before 4 billion years ago. In the very beginning, the early atmosphere of life likely contained water vapor and other chemicals that were released by volcanic eruptions, which were happening all of the time. So nitrogen, nitrogen oxides, carbon dioxide, methane, ammonia, and hydrogen. So there was little to no oxygen in the very early atmosphere of Earth. And just a note about dates, there are just a few examples of dates that you'll need to know for tests coming up. One we want to know for sure is this, that the Earth was formed about 4.6 billion years ago. So I'll always give you, let you know what dates I would like for you to remember. Let me move this out of the way. So again, thinking about timeline of things in the 1920s, two scientists worked together, Oparin and Haldane, to hypothesize that the early atmosphere was a reducing environment. So if we think about what that means, oxidation versus reduction, right? So think back to that. Oxidation, remember, is loss, so losing electrons or hydrogens, and reduction is gaining electrons or hydrogens. And then this continued forward with other experiments. One that was very pivotal early on in the thought processes of how life actually started to form on the Earth was in 1953, same year that Watson and Crick and Rosalind Franklin described the structure of DNA. So Stanley Miller and Harold Urey conducted lab experiments that showed that the abiotic synthesis, so again, without life, so you can synthesize organic molecules in a reducing atmosphere. So this is really interesting. So These experiments helped to demonstrate that organic molecules could have formed in various possible types of atmospheres, but that a reducing atmosphere is a good example of a place where these organic molecules could be created. So let's watch this quick little video about their experiment, and you should be able to describe what their experiment showed.
Stated Clearly presents What was the Miller-Urey experiment? It was once believed that if you left food out to rot, living creatures like maggots and even rats would simply poof into existence. The idea was called spontaneous generation. A series of experiments starting in the 1600s disproved this idea, and in the 1800s, a new scientific law was proposed. Life only comes from life. It's true that rats, maggots, and even microbes are far too complex to simply poof into existence, but in 1859, English naturalist Charles Darwin put forth the theory of evolution. In it, he showed that under the right circumstances, relatively simple creatures can gradually give rise to more complex creatures. Given this information, serious thinkers began to wonder, is it possible that simple life forms actually could come from non-living matter? Not by poofing into existence, but through a natural gradual process similar to what we see in biological evolution. Darwin himself mentioned this idea when writing to a friend. But if, and oh what a big if, he wrote, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, and so on present, that a protein compound was chemically formed, ready to undergo still more complex changes. In 1924, Russian biochemist Alexander Oparin published a book which he titled The Origin of Life. In it, he outlined his thoughts on a gradual progression from simple chemistry to living cells. He imagined the early ocean as a primordial soup, a rich collection of complex molecules produced by natural chemical reactions. In this soup, further reactions could take place, eventually producing living cells. At the time, Darwin's warm little pond and Oparin's primordial soup were really just speculation. They were founded on a good understanding of chemistry and biology, but they could not be considered legitimate scientific hypotheses because no one had found a way to test or observe them. Science, after all, is the study of observable facts and an ongoing conversation about how those facts can be best linked together. Chemical reactions like those proposed by Darwin and Oparin are not expected to leave an observable fossil record. Without either having fossils to examine or a time machine to travel back and observe what happened, how could scientists even begin to study the origin of life? In the 1950s, Stanley Miller, then a graduate student at the University of Chicago, came up with an idea. We could simulate early Earth conditions in the lab and then carefully watch what happens. If you can't study fish in the sea, set up an aquarium. Working with his professor, Harold Urey, Miller designed an apparatus to simulate the ancient water cycle. Together they put in water to model the ancient ocean. It was gently boiled to mimic evaporation. Along with water vapor, for gases of the atmosphere they chose methane, hydrogen, and ammonia. These are simple gases which scientists at the time thought were probably abundant on the ancient Earth. They added a condenser to cool the atmosphere, allowing water molecules to form drops and fall back into their ocean like rain. The ancient Earth would have had many sources of energy, sunlight, geothermal heat, and even thunderstorms, so they added sparks to the atmosphere to simulate lightning. The goal of the experiment was not to create life, but to simply test the first step in Oparin's model. Can simple chemistry naturally give rise to the complex molecules of life? After running the experiment for just 1 week, their ocean became brownish black. Careful analysis revealed that through a series of reactions, many complex molecules had been produced. Among these were amino acids, special molecules of life that we once thought could only be built inside the bodies of living creatures. This was a pivotal breakthrough in science, so significant, in fact, that it gave rise to an entirely new field of research now known as prebiotic chemistry. Scientists don't know for sure if the gases used by Miller really were the most common gases of the ancient Earth. Because of this, many experiments have since been done, showing that the molecules of life can form in a wide variety of environments with different starting chemicals and different sources of energy. Sugars, lipids, and amino acids have even been found on meteorites. This suggests that the molecules of life formed all throughout the ancient solar system and may be forming right now in other regions of our galaxy. Together, these discoveries tell us that Oparin's primordial soup and Darwin's warm little pond could have easily existed in one way or another on our ancient planet. So to sum things up, what was the Miller-Urey experiment? The Miller-Urey experiment was our first attempt at simulating ancient Earth conditions, in this case, the ancient Earth's water cycle, for the purpose of testing ideas about the origin of life. The Miller-Urey experiment is significant for two main reasons. First, though it was not a perfect simulation of the early Earth, it clearly demonstrated for the first time that biomolecules can form under ancient Earth-like conditions. Second, the experiment took what was once mere speculation, the idea that life may have emerged from chemistry, and transformed a portion of that speculation into legitimate, testable science. Many questions remain to be answered about the origin of life, but scientists from many nations and many fields of study are now following Stanley Miller's lead. They're finding ways to turn those questions about the origin of life into testable, scientific hypotheses. Simulation experiments cannot tell us exactly how life formed in the past, but if enough of them are done, they could eventually tell us if it's possible for life to emerge from chemistry. I'm John Perry, and that's the Miller-Urey experiment, stated clearly.
That's a great website. They have lots of good videos to watch. All right. So he mentioned this too in the video that amino acids have been found in meteorites. So that's really interesting. Again, this idea that perhaps pre-biological molecules or components of biological molecules were created in different portions of the galaxy, and maybe they arrived from meteorites from outer space. So that's possible. Also, again, experiments are showing now that these precursor biological molecules can be created in hydrothermal vents as well. So lots of places where these precursor biological molecules, like pieces and parts of amino acids, can be created in these different locations. So what's interesting is that, again, we think about what was the first self-replicating biological molecule, and we're thinking of, would it be either DNA or RNA? Again, lots of scientists have been looking at this, and it seems to be that the consensus right now is that RNA was the first molecule and not actually DNA. So lots of evidence that supports this, because RNA monomers, those nucleotides, can be produced spontaneously from simple molecules. And this happens when these pieces of RNA come together or polymerize when they're concentrated in hot sand, clay, or rock. We also see that, again, we need to take those precursor biological molecules and put them together into protocells. So we know, again, that major key properties of life are replication and metabolism. So being able to replicate, make more cells, and the components inside of cells, and then also that they are able to perform metabolism, so able to take in energy or produce energy within them. And these two properties might have appeared together at the same time in protocells. Protocells typically are thought to have formed from fluid-filled vesicles that likely had a membrane-like structure. So again, the phospholipid bilayer that we think of. And we know, again, that the properties of phospholipids allow for them to create that outer shell for cells. So perhaps, again, precursor biological molecules were being created, at the same time that phospholipids were being created, and then all came together into a protocell. So vesicles, which again are surrounded by phospholipid bilayers, are able to show us simple reproduction and metabolism, and that they're able to maintain an internal chemical environment, which we know again is homeostasis. So again, just more evidence for how things might have occurred in terms of the very first cell that was created. So again, reproduction is a property that we talked about. This is looking at vesicles reproducing. On the outside here is the vesicle boundary, and it has been shown in experiments that if you put RNA or DNA molecules in the environment near a vesicle, that it will actually take up that nucleic acid, which is very interesting. And then this is just showing if you were to, again, in experimental form, have precursor molecules by themselves, or precursor molecules with a particular type of clay that we would find in the early atmosphere, that you're going to have much more reliable vesicle formation with the presence of the clay. So I mentioned this self-replicating nucleic acid likely was RNA and not DNA. And the reason for this is that there are other molecules called ribozymes. So RNA molecules, specifically called ribozymes, have been found to catalyze many different reactions. And what they do is that they're able to make complementary copies of these short stretches of RNA. So if you start reading some of the early data, and these are some of the papers that I read in grad school in my molecular biology studies, that these very early genetic materials might have formed what we describe as an RNA world. So that is typically what scientists describe these early times as, right before life actually existed. So very interesting idea. And again, lots of science that helps to back this up if you were to look into that data. We also need to think about and look at the fossil record. That's going to help us document and look at the changes that occurred in the history of early life on Earth. And again, we have to remember that the very first cells, which again, RNA was likely the nucleic acid, they're not going to leave records for us to find. So fossils are put down in very specific ways by very specific types of animals. And what we see is that these fossils can be dated based on where we find them in sedimentary rocks. So the rocks are divided into layers that are called strata. Strata is the plural, stratum is the singular word for layer. And we see that these sedimentary rocks do have very rich sources of fossils. So again, if you start to dig down into particular locations, you can find fossils, and we're able to date those rocks with different types of radiometric dating. And when we do that, then we can identify the time period that these fossils were created or laid down in the sedimentary rock themselves. What's this? So we've mentioned the Grand Canyon before. Again, Colorado River has carved that out, but you can see all of the layers that have been created over time. So again, when we find a fossil in a particular layer, the closer it is to the surface of the Earth, the younger it is. The older fossils will be found farther down in the sedimentary rock. So again, I just mentioned this. It's the case that very few individuals or types of species have been fossilized, and even fewer have been discovered. So there probably are lots and lots of fossils that haven't been found yet. So the fossil record is always going to be biased in favor of species that were around for a very long time, because there was a greater chance that they would leave behind a fossil. Were very abundant and widespread again, so lots of different parts of the Earth. There are areas that are easier to dig in, more likely to be found in those areas, and if there were more of them. And the other thing is that fossils are generally made by animals that have hard parts, right? So think about bones, skeletons. Those are going to be the types of things, maybe even exoskeletons would be good examples of organisms that would create a fossil. So things that don't have hard parts are unlikely to leave a fossil record. So we might not know about those that are now extinct because we have no record of them. So looking at this process, I mentioned radiometric dating. This is a way that we can actually date and look at what is the absolute age of the fossils. We know that in radiometric dating that there is a radioactive parent isotope. Remember, isotopes are different than regular elements because they have a different number of neutrons. Those parent isotopes decay to daughter isotopes at a very specific and constant rate. And we know this. We're able to measure the amount of parent isotope to daughter isotope that's found within the fossil or the rock itself around the fossil. This gives us a particular half-life number, and half-life is the time required for half of the parent isotope to decay. Radiocarbon dating, again, is used in, also just described as carbon dating, abbreviated that way, uses the ratio of carbon-12 to carbon-14. Again, that can date fossils up to 75,000 years ago. If there's and because there are fossils older than that, we have to use different radioactive parent isotopes. We wouldn't use carbon, we would use something else by looking at the age of the rocks and where we find them in those sediments. So this is just showing you an example of how radiometric dating would work. Again, you can see you start off with a particular amount of that radioactive isotope, and then you have decay that happens at a predictable time, or excuse me, a predictable half-life is known for each of those. So by calculating the ratio of the parent isotope to the amount of the daughter isotope, we can calculate how old the particular fossil is. So when we think about the new types of organisms that were created in the past, we think about mammals. So thinking about us, our group belongs to a group of animals called tetrapods. So again, thinking of 4 limbs. And then what we can see is that these 4-limbed creatures then eventually evolved into mammals and have those unique mammalian features. And we can generally see these features arise through gradual changes over time by looking at different fossils that are ancestors to the earliest mammals. So this is, again, looking at some of the cladistics and looking at the trees that are created for how tetrapods were created, and then again some of the earlier animals on this. So tetrapods, again, you can see created and evolved into reptiles. So snakes actually then lost these appendages. They were left with vestigial femurs, and we've looked at that and we'll talk about that in more detail. You can see again that mammals were a branch off of various other types of tetrapods. So we're going to talk about examples as we go through this and looking at more details specifically about the evolution of mammals that led to the evolution of us, of our hominid ancestors. So looking at key events in the life's history, the geologic record is something that's very important when we're thinking about the process of evolution of life. So geology is an important science, and biologists are able to use geologic record to help them figure out how life evolved. So it's divided into the Hadean, Archean, Proterozoic, and Phanerozoic eons. And the Phanerozoic eon, the very last one, the one that we're in now, includes the last 500 million years, so 500 million years. This Phanerozoic Eon is divided into 3 eras that we'll talk more about: Paleozoic, Mesozoic, and Cenozoic. So these are terms that you probably have heard before. And what we see is that there are major boundaries between these eras that correspond often to major extinction events between the different eras. So we'll talk about those. So you should know, again, the major eons are, first is Hadean, Archean, Proterozoic, and Phanerozoic eons. And then the last eon that's been described, the Phanerozoic eon, is divided into Paleozoic, Mesozoic, and Cenozoic. So this is, again, looking at the oldest at the bottom here. So the Hadean, right, origin of Earth, 4.6 billion years ago. The Archean eon, the Proterozoic, which again is down here. So we can see this is when the oldest fossils of eukaryotic cells are starting to appear. The Archean is when we have our prokaryotic cells. And then again, looking at the origin of the Earth, not much life here, right? The one that we see the predominance of life that has occurred is the Phanerozoic era eon, which we are in right now, the Paleozoic, which is first, Mesozoic, and then the Cenozoic we have here. And again, if you were to think of the geologic timescale in terms of a clock, looking at how this would all go together, and you can see that prokaryotes, right, when they were actually formed, when were our eukaryotes formed, then we have multicellular eukaryotes, animals. This is when things then moved on to land. So at this point, all these other things were in the sea. And then getting all the way to the very last second of the geologic timescale would be humans. So we are very, very young as a species in terms of the geologic timescale. So when we start looking at what were the first single-celled organisms, oldest known fossils that we have found thus far are called stromatolites. These stromatolites are rocks that are formed by accumulation of sedimentary layers that were made up of bacterial mats. So these stromatolites date back to 3.5 billion years ago. So again, if we think of the time scale, right, of when the Earth was formed 4.6 billion years ago. So it took about 1 billion years before the very first things that were alive became prevalent, or at least the very first examples of things that we can see. It took 1 billion years for that to happen. These bacterial mats, again, were prokaryotes, likely also archaeal cells, as we've talked about. They were the only things around for another 1.5 billion years. So it took, again, another 1 billion plus years for eukaryotic cells to actually form. So it's a very slow process. And it is difficult to wrap your head around what billions of years actually look like. But again, those are just some ideas that we have based on the dating system. When we talk about how eukaryotic cells arose and how we went from prokaryotic cells to eukaryotic cells, it had a lot to do with the appearance of atmospheric oxygen. So what we see and what we know based on the evidence that we have is that most of our atmospheric oxygen, and again, in our atmosphere now, we have around 21% oxygen. This atmospheric oxygen was created by biological organisms, which is really interesting. And so if we think about how this happened, about 2.7 billion years ago, oxygen began accumulating in the atmosphere because oxygen was being produced by these photosynthetic prokaryotic cells. So think about these precursor algae cells or precursor cells that would go on to create eukaryotic cells and plant cells. So these oxygen producing cells were emitting the oxygen that they were creating and forming through the process of photosynthesis. So this oxygen that was produced by these prokaryotic cells reacted with the iron that was present in the seas and on the land, and started to precipitate out to form these banded iron formations. And we can see this in these iron-rich terrestrial rocks. So this time period is called the oxygen revolution. So it took place from about 2.7 to 2.3 billion years ago. And because oxygen was going from very little percentage in the atmosphere to up to the 21%, lots of prokaryotic groups started to become extinct because they were not able to live in the presence of oxygen, or some of them even were poisoned by the presence of that oxygen. So those that were able to live through the change from very little oxygen to 21% oxygen, they survived, of course, and adapted to start using cellular respiration, which uses this oxygen. And we know that if you have oxygen and you're able to use it for cellular respiration, the efficiency of your ATP production goes up dramatically. So this allowed for these prokaryotic cells to convert into these eukaryotic cells, and then also allowed for formation of these multicellular organisms as well. So again, if we look at very small percentage, right, of oxygen that was found at the very beginning of when the Earth was formed. Again, our oxygen revolution between 2.7 and 2.3 billion years ago, so dramatic increase. That continued to increase and then leveled off in terms of the present day levels of oxygen that are found in the environment. So again, talking, this is log scale over here, but in percentages, we're looking at about 21% oxygen in our current atmosphere. So that was the prokaryotic cells. Again, and those prokaryotic cells were able to perform photosynthesis, create that oxygen revolution, like we said. And then we have the appearance of the oldest fossils of our eukaryotic cells, 1.8 billion years ago. These eukaryotic cells, we know, evolved to become more complex than the prokaryotic cells. So they started acquiring some of these organelles, like a nuclear envelope, so a place to store the DNA molecule, the mitochondria, again, very important for the use of oxygen in the cellular respiration process, cytoskeleton to help them hold their shapes, endoplasmic reticulum, again, rough ER, smooth ER, which has specific jobs in different cells. And this led also to the understanding of the endosymbiotic theory, which we have talked about before. So this endosymbiont theory proposes that both the mitochondria and the chloroplasts used to live as their own prokaryotic cells outside of these precursor eukaryotic cells. And then what we found was that they were then taken up, not broken down, and maintained for their particular usage. So it became a symbiotic relationship between these precursor mitochondria and chloroplasts. So we see that these endosymbionts are these cells that live within other host cells because there is an advantage for both parties. We talked about this too, this idea that the processes is what we describe as a serial endosymbiosis process, where we think that mitochondria were taken up first. So these precursor cells were actually very similar. They were prokaryotic type cells, and they had acquired the ability to use that oxygen that became prevalent in the environment. And then after the mitochondria were taken up, then these precursor plastids, which became chloroplasts, were then taken up to create eukaryotic cells that could perform photosynthesis and cellular respiration. So we looked at this picture before. So remember, you have your ancestral prokaryotes. So those prokaryotes were early. They again became or started to evolve and create some more complex organelles to give us our precursor eukaryotic cells. And then what we saw was that these precursor eukaryotic cells engulfed or ate up these aerobic, remember, bacterial cells that are able to perform cellular respiration aerobically, meaning they're using oxygen. They took those up. Some of these, again, continued to evolve and became our ancestral heterotrophic eukaryotes. So again, heterotroph meaning getting energy sources from somewhere else. So those would go on to become animal cells. There was another group of these precursor eukaryotic cells that actually still had the mitochondria, but then secondarily engulfed a photosynthetic bacterial cell, those precursor plastids, which became the chloroplast. So these eventually evolved into our eukaryotic cells that we would find in plants. So again, the mitochondria was taken up first, and then the chloroplast was taken up next. And these two types of eukaryotic cells were maintained over the evolutionary process. So we also looked at this before too, this idea of what is the evidence for this process of the endosymbiont theory. And these are the pieces of evidence that help support this. So we see that the inner membranes that we see inside of our eukaryotic cells are very similar to the plasma membranes that we see in prokaryotic cells. We see that these organelles, both the chloroplasts and the mitochondria, have DNA inside of them, and also that they're able to reproduce those organelles. The reproduction process of the organelles themselves, the mitochondria and the chloroplasts, and the DNA inside of them is very similar to how this would work in prokaryotic cells. This, again, these organelles maintain their own DNA. So very strange for organelles themselves to have DNA unless they used to live on their own. And then they also do have ribosomes. So there are ribosomes that are found in these organelles, which look very similar to ribosomes that we would find in prokaryotic cells as well. So again, all evidence that helps to support this theory that we started off with a ancestral prokaryote, took up a precursor bacterial cell that became mitochondria. This population went on to make the heterotrophic animal cells, and then another subset of these organisms went on to pick up something which was a precursor photosynthetic bacterial cell that eventually became the chloroplast. And then these eukaryotic cells became photosynthetic. So they were able to perform photosynthesis, but then also cellular respiration as well. So looking at the process of going from single cell to multicellular organisms, again, when we think of why this was able to happen, really it was because of this endosymbiont theory. So this idea that these precursor eukaryotic cells picked up what would become mitochondria. So the ability to use oxygen is very important and allows for greater ATP to be produced from just one glucose molecule. So the evolution of these types of cells that had the mitochondria and the chloroplasts allowed for a greater range of unicellular forms. So again, what does the outside structure of this look like? And then we had a second wave of diversification that occurred when multicellularity evolved and gave rise to things like algae, plants, fungi, and animals, which is really interesting as well. So we'll talk about this. All right, we're going to pause here, and I will pick back up in another video. Thanks.